Well, hello everybody and welcome to another episode of GUcast. This is Declan Murphy, urologist here at Peter MacCallum Cancer Centre in Melbourne, joined as ever by my co-host, Dr Vinay Prasad, also urologist here at Peter Mac. Good morning, Renu.
Good morning, Declan. It feels like New Year's Day.
Wow, you're all smiles. Why are you all smiles this morning?
I don't know. Maybe because we've come out of lockdown?
Yeah.
We have come out of lockdown before, only just to go back into it again, but this time it feels
It feels like it
definite.
might be the last time. Yep.
Yeah.
So for those of you out there who've listened to us moan about being in lockdown and hauling ass in here at 6 a.m. to talk to international guests in the midst of curfew and restaurants closed and kids homeschooling, well, today is Freedom Day, hooray, or so we think anyway. Well, we'll see. It seems like we might actually get away with it this time because we are vaccinated and hopefully we will. And our guest, by the way, this morning knows a lot about vaccinations and COVID, but we don't really want to talk about that today. And we'll get to our fantastic guest in a second. But let's introduce our other fantastic guest and welcome back a friend of the podcast, Professor Michael Hofman, nuclear medicine physician extraordinaire here at Peter Mac. Michael, thanks for coming back in.
Great to see you.
Good morning, Renu. Good morning, Declan. Pleasure to be with you again.
Well, are you feeling all full of freedom this morning?
Very full of freedom.
Kids happy, wife happy?
Kids are all going back to school today.
Oh, really? They get back
all
today?
on the bus.
Yeah. Amazing.
Wow.
Mine are back next week, but it is good, isn't it? It does feel, the city does feel finally like it might actually break through this time. And
My daughter's having a sleepover on Saturday night. She's so excited.
it's crazy, all these things you took for granted.
It's insane. There were people at the hairdressers like at 3 a.m.
Really?
Yeah, just,
Yeah, we're having a birthday party in the park, or it was supposed to be in the park, and now it's down in someone's backyard this afternoon with a swimming pool and so on. First time in months and months the kids can see each other. But there you go, that's all good. So the mood is good here in Melbourne today. And now we're going to cross to San Francisco to have a chat with our fantastic guest, Dr Vinay Prasad, who's joining us from UCSF today. Hello, Vinay, and welcome to GUcast.
Thank you for having me. It's a pleasure to finally make an appearance on the show.
Well, look, it's absolutely great to have you. We better introduce him formally, but I'm
right.
sure a lot of our listeners know Vinay already. Dr. Vinay Prasad is a Haematologist Oncologist at UCSF and Associate Professor of Medicine at UCSF in San Francisco. And he's very well known as an eminent oncologist, of course, but he's also very well known to those of you who like things like podcasts or YouTube channels. He runs a fantastic podcast called Plenary Session. We'll put a link in the show notes, which also broadcasts now on YouTube as well. And he's written a great book called Malignant. So his interests are not just in oncology from a clinical point of view, but he's very interested in health policy and in research methodology and calling out bad research and bad policy. Is that a reasonable summary, Vinay, of some of your interests outside your clinical practice?
You have the diagnosis accurate, the good doctor.
Well, look, it's great to have you. You are a fantastic podcaster and we enjoy supporting you. We're putting a link to your Patreon as well. I think it's really valuable when clinicians take time to do this sort of stuff for new, and it's good to support these type of endeavors. So we support you on Patreon and we have done for quite a while.
It's always, I like the stories behind how podcasts start. I mean, we started because we couldn't travel to conferences anymore and we wanted to still connect with our international friends. And Vinay, what about you? What made you start podcasting?
That's interesting. I didn't know your origin story, but I also like these stories too. So my story was, at the time I was working in Oregon Health and Science University. I was based in Portland, and I had a pretty lengthy commute. I had about 8-mile bike ride. I would bike either way on this old trail. It was pretty safe, and I would listen to podcasts myself. And I started listening to, all the common things, This American Life and Freakonomics and, all the big national, international shows. Eventually, I stumbled onto this podcast that was done by lawyers for lawyers. It was about the Supreme Court of the United States. And, I don't know a lot about the Supreme Court. I'm not a lawyer, but I just listened to it and I found myself really just fascinated by it. They didn't dumb it down. They talked about it for like a colleague, a professional colleague. And I thought to myself, I wish there was something like this in oncology so I could listen to people talk about clinical trials and how they think about it. And I looked and, we had a couple things at that time, maybe ASCO, some of the journals had a podcast, but without getting too much into it, I think that the quality wasn't great. It was always like a phone call that was poorly recorded and you could hardly understand what they were saying. So I thought, it might be nice to actually try to make a real oncology podcast. So I launched in 2018. I think in the summer. And, it just kind of goes from there. I will say of all the things I've done professionally, it's probably one of the best things I've done because I get the most positive feedback from it. It's not like Twitter. It's not like writing papers even where a lot of times people don't read it. You actually get notes from people who say, this really resonated. So I really enjoy doing it. I feel like you must feel the same.
Yeah, and look, and I think that's one of the points of difference. And podcasting was growing. I think that's what we've observed before is, people were having mirroring apps in their car, or you're out for exercise, and Spotify invested a lot in. And so podcasting was growing and growing, but we struggled as well to find a good podcast. And the problem is as well, I think you'll share this view, is that a lot of it tends to be either industry dominated or constrained by it's a journal or it's a publishing platform and so on, and not really clinician led. And that's one of the things we love about your rather disruptive attitude is you do challenge stuff really well with no constraints. Yeah. And we have played clips from your previous podcast before, but that brings us to why we've invited you on today, because you did a great podcast recently following the publication of the VISION trial in the New England Journal of Medicine. And VISION, as we've talked about on this podcast before, is an important trial for those of us interested in radioligand therapy in prostate cancer. And that's why we've invited Michael back into the studio as well today. And on the podcast, Vinay, as ever, was quite critical about elements of the study design and about the control arm. We enjoyed listening to it, Renew, I think it's fair to say, but we felt that perhaps we should have a go and see would we balance off some of the criticism. So we invited Vinay on to talk about that.
Maybe just some background about VISION. How did we get to VISION? Because we at Peter Mac started very early in this field. We did PSMA PETs back in 2014 and the first lutetium PSMA therapy in 2015. And we did that as part of a phase 2 trial, which was not industry sponsored. It was an investigator initiated study. It was when the IP of this compound was not really well known and a small company owned it, and we took it up with no grant funding and did this 30 patient study that was published in Lancet Oncology back in 2018. And that study was really when industry got interested and changed hands several times, and that was the birth of the VISION trial, that little 30-patient Peter Mac study.
Well, look, while we have you, Michael, will you, just for those who may not be familiar with VISION, can you give us like a Twitter-sized summary of VISION before we go on in and talk about? We'll put the links into more detailed presentations on it before, but tell us a bit about VISION in summary.
I was going to say Vinay might do that better than me. I can do therapy.
No, we want him to critique it. Just give us a summary. What was VISION all about? Just a rough overview.
Rough overview. It's a industry-sponsored large phase 3 multi-center randomized trial comparing lutetium PSMA 617. This is a radioactive small molecule for targeting metastatic prostate cancer, compared to protocol-defined standard of care in a 800 and something population, randomized 2-to-1. Primary endpoint overall survival, and successful in that endpoint with around a 4-month improvement in OS, and also improvements in quality of life that I think we should come to in this podcast because that's really important as well.
And the timing of VISION was important, wasn't it? Because just before VISION results were released, therapy was released, and so it led to a lot of comparisons between the two trials, which I'm sure we'll touch on in this in this talk as well.
Yeah, exactly right. And in Vinay's podcast, he focused on the VISION trial and important pivotal, registration level study in the New England Journal of Medicine, big OS benefit, et cetera, et cetera. So, and very much welcomed by many people out there, including those of us with an interest in not just prostate cancer, but radioligand therapy. However, Vinay, you certainly, as you tend to do, picked up on a few points in it. And in fact, of course, we share actually some of your interesting critiques, but I want to play you, if you don't mind, one of the things that you picked up on, which you often do in these trials, especially industry-sponsored trials, and it's really important to do, is the control arm. What is the standard of care? Is it an appropriate comparator? Is the control arm handicapped maybe by being a weak link and so on? And here's what one of the comments you made about the control arm.
This means this is a population where 50% of people could have gotten cabazitaxel, which is probably what a lot of people would do in the real world. Why? Because the CARD trial, where cabazitaxel tested against an androgen signaling inhibitor, has a survival benefit of a couple months. So this control arm, they're deprived of a standard of care therapy. Now, imagine I was a patient enrolling on this study, or a doctor. Well, a doctor, I wouldn't have a lot of respect for myself to enroll on this study because it is an unethical, egregious, and horrible control arm.
So, Michael, what's the big deal with the control arm? So these are men with very advanced prostate cancer. They've progressed. They've had a bunch of drugs already. They're nearly out of steam, aren't they? They're randomized to get ongoing standard of care versus this new radioligand therapy. So the point that Vinay is making is that that's not a good standard of care, is it? So before we ask him to comment on it, what is the problem with that? They were being offered certain standards of care, but they were being deprived, as Vinay said, other standards of care.
Yes, I think that's true. And the main standard of care being deprived is cabazitaxel. And the authors of VISION may argue, in fact, you had to have progressed after cabazitaxel or be unsuitable for carbazitaxel to get on the trial. But as Vinay pointed out, around over 20% of men received carbazitaxel in the control arm afterwards.
Yeah.
So they must be suitable by definition. So it's a little bit murky. I think that's the main criticism. Also, if you've had Enza, Abbey, and then you go back to Enza again, that doesn't happen in Australia at all. I don't think anyone does that. Yeah. Do people really do that in the US? I'm not sure.
No.
So the crossover. But I'll put it to you, Vinay, what about if they had a placebo as the control arm in this study? So the same design, but the control arm being a placebo injection. What would you think about that?
I would find it, placebo, but then no other standard of care, or you're allowed to use cabazitaxel?
No, just
I think
placebo, straight placebo.
That'd be no good.
Yeah, I would find it problematic, because I think that this is a population that they are fit, they're eligible, they can get life-prolonging therapy.
What about if they were
life-prolonging therapies, right, that are available to them?
What about if they were post-carbazitaxel?
Then I think now you're getting intriguing. Now, so I think that there is a population at the tail end of, they've literally exhausted every single proven therapy that you could randomize, I think, ethically to placebo. That would be a different study. It would be biting off a different chunk of market share, much smaller market share. But I think there is that space at the end of all treatments.
But you still might have the same problem. You'll have some men that are medically just unsuitable for carbazitaxel. They're 80 years old. You can't say can't go on this trial because you haven't had carbazitaxel, because they're not suitable. So it gets a bit tricky.
But that's the problem with this trial,
they
though,
may
is
not be
that
suitable for lutetium too. That's the other question, right? Yeah.
Yeah, but the thing is, this group of men probably were suitable. Many of them were. 40% of them had got cabazitaxel beforehand, but that was Vinay's point, is that like half these guys were handicapped because you're randomized into the control arm, and then you're getting nothing. You're getting steroids or whatever. You're getting more Enza, which is no good, but you're not allowed the two very active things, certainly in the US, that would have been available, cabazitaxel or radium-223 for certain men. So the fact that neither of those active therapies with an OS benefit in this setting already established were allowed is a problem, isn't it? Because I know we'll talk about the CARD trial in a second, but already we had evidence that cabazitaxel improved survival, but they weren't allowed to have it because it would have to be combined with standard of care in the intervention arm as well, I think.
But do you know why they didn't choose placebo?
Well, it would have been, nobody would have.
No, if they made it post-carbazitaxel.
Yeah.
Because you can't do placebo in a radiopharmaceutical trial, because you cannot blind anymore, because with this device, a mobile phone, did you know you can get a radiation monitoring app? What you do, I'm not joking, this is serious, is you get black tape and you put it over the camera, and photons from your lutetium therapy pass through the black tape, and your CCD, your camera, detects it on your phone. So there's radiation detectors on your phone. And you can just download these apps for free. And then I could put it in front of you and detect whether you're in the lutetium arm or whether you're in the placebo arm, and you can't blind it.
Yeah, it is
Can I
progressive.
comment about that real quick? I agree with Michael's point that that it is an unblindable product. I think it is unblindable. You can have a placebo bag or whatever, but you can easily use a Geiger counter and figure it out. You can, go to your local store or whatever. But my point would be that why would one want to blind? There's certain endpoints that are bias susceptible endpoints that can be affected by your knowledge as to whether or not you received the product. But if you really did, in a last ditch, last line effort, lutetium one PSMA versus, best supportive care or placebo or observation, and you had an OS benefit, no one would think that could be influenced by the placebo effect. It's a real OS benefit. So, many trials are in fact open label. And so I don't think that's the issue. The blinding, I don't think is the issue. I think there are other issues that we could talk about.
The main issue is that, as you say, they are handicapped. They're not allowed to have cabazitaxel or other things like radium-223. So this is a really big problem, because we know that there's OS for those. And yes, the CARD trial came out afterwards just pointing out that, cabazitaxel is better than what many of them did get, which is ongoing AR-targeted therapies, which by definition they'd already failed. So I think, number one, we should all accept that that's a genuine criticism of it. And I still think that the authors should acknowledge that. In fact, we of course, have talked to the authors, who we know well, and we congratulate them on this. But here's what Oliver Sartor had to say to us on the PCF webinar that we posted on GUcast a few months ago, when we talked about the control arm.
Chemotherapy was a potential active compared to the control group. However, we weren't really sure what chemotherapy was going to do on survival. And by the way, when we designed it, we did not have the CARD trial. We just knew that we got to be a little bit concerned. But one of the things we did was allow 2 lines of prior taxane chemotherapy. So there is no third-line therapy. So by allowing the first 2 lines to proceed as inclusion criteria, we felt better about excluding chemotherapy in the control arm. It turns out that about 40% of the patients had cabazitaxel and docetaxel. So this would have been a 3rd line of chemotherapy, and that's not suitable. Another thing really important, if you look globally, very few patients get 2 lines of chemotherapy, and virtually no patients get 3 lines of chemotherapy when you look at a statistical basis. Look at the real-world evidence that we've garnered. People like Dan George and I have published real-world evidence. Most of the people, and this will be shocking to some, most of the people never get docetaxel before they die.
What do you think, Vinay?
Okay, I guess a couple of thoughts. So I guess first, the last point first. This is a new talking point. I mean, it's being promulgated by the industry, and everyone is saying it in every setting, which is they're saying in the real world, patients are not getting 2nd line, 3rd line therapy as a justification for not providing that in a randomized controlled trial in the frontline setting, that post-protocol therapy. This is problematic in a number of ways. One, they're often looking at sites and locations that simply cannot afford those post-protocol therapies. Number 2, the people who are unselected, all comers in general clinical practice are often older and frailer than cancer patients on pivotal trials. You cannot compare real-world utilization in Eastern Europe against what would happen to someone who is fit enough for the trial you're enrolling in the United States. They're going to have much higher rates of subsequent lines of therapy. They're going to have much higher rates of different things. And so this is just a tactic I see employed in many different tumor types, arguing why it was justified to have sort of inferior post-protocol care, which will affect your OS. The other point he is making about lines of therapy, I guess I was confused because there's at least 60% of people who hadn't had cabazitaxel. Yeah. We talk about the fraction of people who got cabazitaxel after this trial ended. It was about 20%, I think, of all who were randomized. But among people who got some medication, I think it was about 2/3 of them got cabazi. No matter how you want to slice it, I think it is clear that probably many of the people who are put on this study could have tolerated cabazitaxel. They would have lived longer had they gotten cabazitaxel. Very likely they would have lived longer had they gotten it earlier rather than after the protocol. And that would have made the benefit of lutetium get a little bit smaller. But could I say one thing about how I think about this whole space that might be helpful? I mean, in cancer medicine, there's just 2 things we're trying to think about. One, does this drug have activity? There's no doubt in my mind, lutetium has activity. It's an active drug. By active, I mean it shrinks tumors. And active drugs, it's great to know about active drugs because active drugs are the backbone of successful cancer treatments in every single tumor type. The active drugs are great. We all want active drugs. The second question is, once you identify an active drug, where do you use it in your lines of therapy? Do you give it early? Do you give it in the middle? Do you give it at the end? And that's when you need a randomized trial like VISION. You need to test giving it at some spot versus the best available care you would give if you didn't have access to that drug. Yeah. So that's the philosophical question. So, I have no problem with this drug. I'm confident this drug is active. It's quite active. In fact, it's probably a good drug. It will have some role in prostate cancer, but is the role here? And this trial, I think, stacks the deck to make it say, yeah, it's right here. But in reality, they're doing all these things in control arm that make it an easy win. It's unfair.
And that's one of the main differences between vision and therapy, isn't it, Michael?
Yeah, so we designed the therapy study in 2016 together with the ANZUP Cooperative Clinical Trials Group. Again, this trial was designed when nobody had heard of lutetium PSMA, the therapy trial. No one had any interest. Novartis didn't have the asset. Even Endocyte didn't have the asset, so there was not much commercial interest. So this Australian Cooperative Trials Group got on board, and I had the pleasure of leading that trial, and it was run at 10 centres around Australia, published in The Lancet in March this year, and it was a 200-patient study of exactly the control arm that Vinay describes. Exactly. A very clean study, post-docetaxel, 91% post-enza-abi, and lutetium PSMA 617 versus cabazitaxel.
Yeah, and so it is. What do you think, Vinay? I'm not sure. Did you get a chance to have a look at it? But it is at least.
I reviewed it. Hofman et al.
Yeah, there you go. What's your take?
Michael's pet name here.
Okay, here's my sense on it. So 1, it's a good study. Hofman et al. It's a good study. It's a good comparator. Yeah, it's a good comparator. It's a good study. But I will say one thing that I think you'll find that may be of interest. The primary endpoint of this study, and it was powered and designed. It was a phase 2 study to assess an increased response rate. And lo and behold, they hit that. It was actually a really nice power calculation. Michael can talk about it. They hit it beautifully, 66 versus 39% response rate. By the way, one point we're making is what was the response rate on the control arm in Vision? I think it was a 3% or 4%, proving that they're giving really ineffective therapy to that control arm, right? It was like essentially no responses. But in this trial, it was a good study. It was powered for a response rate therapy. It found it. It can conclude, I think, rather solidly that it has a better response rate. Now, the next question is, can you hang your hat on a PFS out of this study and an OS out of the study? I'm always reluctant in phase 2 trials to use those other endpoints. I think we ran into trouble with Lartruvo and some other examples in oncology. So I would just like to see therapy as a phase 3. Just run it in a bigger study. And then you've got, I think, a very satisfying registration study for any drug regulatory agency.
I don't know that anyone's going to repeat the therapy study in a larger cohort. So I think now we have a mishmash. We've got Vision. It gives us valuable data, even though your criticisms, we agree with them. But I think, you can kind of blend a bit of therapy and Vision, and you can get halfway between. And I think what's also clear from Vision and therapy is quality of life. In the therapy study, multiple quality of life parameters favouring lutetium PSMA over carbazitaxel, less fatigue, no neuropathy, less. We can go through the list. There were heaps of quality of life parameters hugely favouring lutetium PSMA and none favouring carbazitaxel. And at ESMO this year, the quality of life data from Vision was also presented. I don't know if you saw that, Vinay, but it's quite compelling. Again, bad control arm, as you say, but pain. I mean, the Kaplan-Meier curves for sort of delay in onset of pain was so far in favour of lutetium PSMA, and that's what we see clinically. We have patients with bone pain, gnawing at them at night, and they have a dose of lutetium PSMA, and a few days later they're starting to wind back their opiate analgesics. 2 weeks later they're off all their analgesics, and we don't cure them, but their quality of life improves. And you've got the mantra of there's only 2 things that matter: overall survival and quality of life. And I think we all agree with you. Like RPFS, PSA, PFS, PSA response rates, they don't matter. But I think if you look at Vision and therapy and you look at them both combined, you hit those 2 points pretty compellingly, both OS and quality of life. And even if you don't do OS, even if you're right that carbazitaxel is about equivalent, so you're not prolonging life, you might be right about that. We don't know because we don't have that phase 3 data. You would still show that improvement in quality of life. We know that. So your quality of life would be better.
And that's still something to take away,
And just on it, OS was a secondary endpoint. It's not powered for that, but you will be reporting OS in therapy at some stage.
Correct. So therapy has OS follow-up to 31 December this year. And when we hit 31 December, our statisticians will hit the play button on OS. So we may have that data. Or we will have that data. We'll present it either way.
Yeah, so it's a mishmash, and that's one of the things. It's an academic study. It doesn't have the spectre of we need to get this registered by the FDA. We're in discussion with the regulators, etc. But it's hard to interpret a mishmash from a quality of evidence point of view, isn't it?
So Vinay, help me out here, because Vision was a registration level study. The FDA got involved. They reviewed the protocol. Our little therapy trial had no regulatory oversight. We just had medical oncologists, urologists, radiation academics in a room designing this trial. And we chose carbazitaxel as the control arm. But, Vision was the registration trial and they were guided by the FDA. So I think Mike Morris will say Vision was a registration study. We did it to enable men to access lutetium PSMA, and we sought the guidance from the FDA as to the appropriate way to do that in the fastest mechanism, and this is what they came up with.
Is the FDA stamp a stamp of quality these days, or what does it mean?
I don't know. We don't live in the US.
I struggle with it. Yeah. I mean, I think their rubber stamp might be broken with the way they're approving products. I guess I'd say just a few thoughts about some of the things you're talking about. I mean, I think, like I said, this drug is active, so it'll have some role in prostate cancer. The question is always where and what sequence. The only thoughts I had about quality of life, I think you're right. The two things you always have to look at, and I haven't looked at too closely with quality of life, is there's often a lot of attrition in how many people actually fill out the forms. And we've done a lot of work trying to model assumptions around censoring of quality of life. The second thing is like how long they follow quality of life. You always want to, I think what you really are asking is if you get a sequence of drugs, lutetium, PSMA, and then cabazitaxel potentially for the fraction of people who can still tolerate more, or cabazitaxel and then lutetium, PSMA for the fraction of people who can still tolerate more. You really want to look at quality of life over the duration of the cancer journey. And we've done some papers where we show that actually quality of life is often presented just in a very short snapshot. It's really sort of like an early quality of life and not sort of a longitudinal quality of life. But I don't disagree with you in the sense, I think also radium, I've seen great responses from radium for people with really bad bone pain, which is another reason why it should have been a control arm. But you're right. I mean, an active drug will generate, I think some people will just feel better because they have disease pressing up against something you don't want to press up against. The point about the FDA, I would say this. I mean, it's a captured agency. It's really captured in so many ways. It's captured in, when people work there, they work there for short terms mostly, and then they predominantly go to the biopharmaceutical industry when they leave. The incentives at working at the FDA are to play nice, to play ball. In fact, that's kind of the incentive in all of our profession, to play nice, play ball, and go with the flow and not ask tough questions. I commend you all because you guys actually do ask tough questions to some people, but I think that's rare. And so the FDA's, I imagine the discussion was that the company tried to push the FDA as much as they could to get the absolute most favorable design that they still won't reject. But I do think there's some problems with vision that even the FDA might not be able to overlook. And that is when you get 50% of people initially assigned drop out, and then even after that protocol amendment where they sort of educated them to stay on the control arm, there's still a very high rate of dropout. The only precedent for that in my mind is quizartinib in AML. It went to the Drug Advisory Committee. And when you get a lot of dropout, the FDA treats all the results with a lot more uncertainty because they only represent people who didn't drop out. People who drop out are different than people who don't drop out. They tend to be wealthier, well-connected, more options. They know their power in the world, and they can go get different care. And so you create some imbalances that sort of subvert randomization. Randomization, of course, the beauty is you balance known and unknown covariates. But when you have selective dropout, a guy like me is the kind of guy to drop out because I know I have connections. And so people who may be more vulnerable, less likely to drop out. And so that creates some problems. So we'll see what FDA does with this.
Well, look, that's one of the other really, really important points. And you had a great, can I call it a rant? Is that rude to the guest?
Yeah,
it's a rant.
It's a rant.
We love your language.
Let's play it.
I know. Let's play your tip.
Also calls it a rant.
Exactly. And for those who don't know, so obviously randomized trial, there's the intervention arm with lutetium and the standard of care arm, and you expect a little bit of dropout. And Vinay and colleagues have published quite a lot about this over the years, and he refers to it in his podcast on the topic. But here's what he had to say about the dropout in the control arm.
After the trial started, a high incidence of withdrawal from the trial was noted in the control group and at certain sites, probably sites where they have other options, and was attributed principally to patient disappointment. That's not so good. You can't run a trial with a control arm so lousy that patients are literally disappointed and withdraw consent when they match into the control arm. After discussion with regulatory authorities, we implemented enhanced trial site education measures. Indoctrination on March 5th, 2019 to reduce the incidence of withdrawal. So we tried to indoctrinate patients to stay on our lousy, unethical control arm. That's what you did? How is that acceptable? And you discussed it with regulators. Did they approve of this? Or did you just mention you might do this? This is disgraceful. I find that very disgraceful. Look, the percent of patients in the control group who discontinued the trial without receiving the randomly assigned treatment was 56% of patients before the implementation of these measures. 56%? Get out of here. That's astonishingly high. And I'm about to show you some empirical data that shows just how horribly high that is. Patients are sniffing out that you're giving them a dilapidated, delinquent, and unethical control arm. And after you did your brainwashing sessions on them, you dropped it down to 16.3%. How is this ethical?
And it goes on. It's such a great rant.
Me.
But Michael, we heard about this early in the study because apart from therapy and the original phase 2, we have multiple other prospective trials running in lutetium. We have a very research-oriented approach to this, and we're happy to talk about those. But we heard rumours very early on that there's a problem in the control arm early on in the recruitment, and we were hearing this. Unsurprisingly, people were dropping out when they got randomised to standard of care. So that's definitely an issue, Michael. Before we go back to Vinay, and because we want to talk about the methodology of that and the fallout from it. But your thoughts on the dropout initially, and are there lessons to learn from clinical trialists and equipoise, all sorts of questions? Why half the people go, no, thanks very much, I'm out of here.
Yeah, look, I think it is very problematic, that high dropout rate. It's a marker of the trial design and a lack of active agent in the control arm, and that's probably cabazitaxel not being there. But there is one nuance here, and it is that these radiopharmaceuticals do follow a development pathway that's different from medical oncology drugs. And many medical oncologists may not be aware of this. But, you know, these radiopharmaceuticals, they don't tend to evolve from industry, and they don't even tend to evolve from phase 1 studies. So there is no phase 1 study of lutetium PSMA 617, quite remarkably, and that's because its first use was compassionate access treatments in Germany. And very German style, and in some parts of Europe and also in Australia, is that these radiopharmaceuticals can be made in hospital radiopharmacies, and then they're given under compassionate access legislation. And since you can see what you treat, and it's got analogies more to external beam radiation, you give the drug, you put the patient on the gamma camera, we can do whole body fancy dosimetry scans, find out tumours getting 120 gray, normal tissues getting 3 gray, we get the safety measures from our images, then we start using it. And this happens in Germany a lot. That's how lutetium PSMA evolved. So when the VISION trial opened, not in the USA, but in other countries, lutetium PSMA was already available. And word had got around. If you were a patient and you were logging onto your patient forums, you already knew the results of the VISION trial. The patients were describing, my pains disappeared, I had 4 weeks to live, and here I am 9 months later, the exceptional responders, because we do see those. We had our data from Peter Mac, our image of the year, showing these exceptional responders to lutetium PSMA. So no one doubted it was a highly active therapy. But if it wasn't available, you would still go on the trial and try your luck. The problem in the control arm for VISION was patients had options, and they could fly overseas. It was pre-pandemic. They could fly to Germany, pay their few $1,000s, get their lutetium PSMA. So it created this complexity that I don't think exists in the medical oncology world as much, because before a drug is approved in medical oncology, it's really hard to access that drug. It's almost impossible. Whereas in the nuclear medicine world, it's a little bit different.
Yeah, but so I'm still puzzled about this. So the standard of care arm didn't change to address the dropout rate. I might have thought, okay, one solution here is to say, well, to restore equipoise and balance, we're going to allow cabazitaxel, surely. Okay, that's the obvious. But that's not what happened, Vinay, is it? So instead, I don't get this. They went to the regulators and they said there was an education process. I don't get it. But what can you please expand on the delinquent dropout rate?
I mean, I guess I don't know what to say other than we've done sort of an empirical work where we look at many trials where this data was available, so we can see differential dropout rates. And I will tell you from that work, on average, there's about a 0.5 percentage point more dropout on control arms than active arms across all cancer medicine. And that's to be expected, because as much as we blind and placebo-controlled products, you got some cancer drug because you get hand-foot syndrome, or you get, a rash, or you get some side effect that kind of clues you in you're getting the drug. And people get placebo initially when they kind of get the sense they're getting placebo. There is a slightly higher rate of dropout. But this trial is off the charts. I mean, this has never been seen, I think. And I kind of showed in my YouTube video a graphic of it. It's really sort of off the charts. It's never been seen. I think you're spot on. I think that does reflect, and Michael's point is actually astute, and I don't think I knew all that. But that's a very good point, that if you're really well connected, you have all these other options. So why would you want to be disappointed when you don't have to be? But it is a problem for the trial. It actually fundamentally, with that dropout, you just thwarted randomization. I mean, you're randomizing, but then all the rich people can fly to Germany are dropping out. So that's not random anymore. That's a real covariate that you've introduced. And the other thing is that the remedy was not the right remedy. It was the corporate remedy, which is how do you salvage this trial and still compete for registration, rather than the clinician patient-centered remedy, which is we got to rethink this trial and maybe we need to just take therapy and scale it up, and that might be better.
Vinay, you've done a lot of work on, a lot of studies on control arms in clinical trials, which, underlies your criticism of control arms in many of these trials. And in fact, when it came to the VISION trial, you compared it to the PROfound trial in that it was profoundly bad, the control arm. In some of the data that you showed in your YouTube video, you commented that 1 in 5 control arms are actually suboptimal in clinical trials. Why is that?
That's because of the US FDA. I mean, I think the US FDA, I guess I'd say a couple of things here. One thing is, why do we do randomized controlled trials at all? Well, we do it because we want to get a more truthful claim, like the product improves outcomes, than just observational studies. And that's true. But just because it's called a randomized trial doesn't mean it's a good randomized trial. You got to test the new thing against the thing you're actually doing. And you also have to, if you use PFS as the primary endpoint, that's enough. But if you use PFS2 or overall survival, all of the things that happen after the trial end matters a great deal. And you all raised this point very well with the Pembro adjuvant RCC study with Dr. Churi. And all of those post-protocol things matter. Well, what we have right now is an environment that the FDA has two roles. One role is to make sure products that come to the market are safe and effective for American consumers. But the other role is to kind of empower the biopharmaceutical industry in the United States to be able to charge significant revenue for their products. That is, in fact, one of the goals. And the former commissioner, Scott Gottlieb, pointed out that they often forget the second goal. But in the last 15 years, 20 years, we've seen a big swing to that. So the FDA has empowered companies to sell their products. And one way to sell your product is to say, ibrutinib, it's a great new drug for CLL, but it's hard to beat bendamustine, Rituxan, and FCR. It's easier to beat chlorambucil, so let's let you test against chlorambucil. In kidney cancer, every single frontline study of the double is against sunitinib. In the second line setting, they all go against everolimus. They pick the one thing they can just keep beating up. This is a problem across the space. Actually, those are not as egregious examples as in some of the other tumor types. But I think the FDA has the legal authority to halt unethical control arms. I'll give you one example. There was a trial many years ago in melanoma. Ipilimumab had already shown a survival advantage in combination with GP100 over GP100, and it was the standard of care in the US. They wanted nivolumab to get the market share. They didn't test against IPI. They tested against dacarbazine. The FDA had a lot of problems with that study because dacarbazine was at that time, I think, an unethical control arm. It was not what we were doing in the States. The company ran the trial, and this was one of the few times the FDA they threw away the control arm and they approved nivolumab with an accelerated approval based on response rate. They didn't give him a full approval based on randomization. So that was one way they pushed back. But in the years since then, the appetite at the FDA to approve products has grown. And I think that I'd be surprised. I'm curious what they do with vision, but I think that's the core problem. It's the FDA.
I just want to touch on the control arm and the fact that it was done in the USA, because Declan didn't get a clip of another one of your rants, but I noted it down, so I'll try my best.
Yeah.
You said,
are you going to do my voice?
Yeah, that's right. He said,
you have to have all the gestures and it's all hair and teeth.
The places where this control arm reflects the standard of care, they can't afford your PSMA scans. They can't afford your lutetium PSMA. This only applies to this country, the USA.
Yes.
So I guess my point there is that there's this problem in all cancer medicine, many trials, which is the following. They do a trial of Pembro frontline or Pembro adjuvant, and they go to countries and they run that trial. And then the control arm, when they progress, they don't get pembrolizumab because that's not the standard of care in the country. So I was like, who does this trial help? It doesn't help that local site because they can't afford pembrolizumab in the second line setting. How are they going to afford it adjuvant? And it doesn't help us in the US because we're giving pembrolizumab in the second line setting or first line setting. So do we really know adjuvant is preferable to that? So it kind of sates neither party, but that's the nature of commercial research organizations.
Yeah, that's interesting. But is that exactly true with PSMA? I think.
Oh, maybe. Okay.
Yeah. This is where we want to pick you up, Vinay, because you like the facts.
Yeah.
And the facts are that if you go to third world, not to developing nations like India or South Africa, you can get lutetium PSMA today, and it's really cheap. In fact, in South Africa, people get lutetium PSMA in preference to enzalutamide because they can't afford enzalutamide. In a fee-for-service practice, they go to their oncologist and they go, Enza is this per month and lutetium PSMA is this per month, and they buy the lutetium PSMA because it's cheaper. And that's happening actually today, and it's happening in India as well. And then it's happening with PSMA PET scans as well. In India, you can get a PSMA PET scan for $200 US.
Yeah. And in Australia, Germany, many European countries, this is totally replaced conventional imaging because PSMA is cheap as chips, because the stuff there's no IP. And the same with lutetium PSMA. The 617 now has some IP, but PSMA INT doesn't. So we have widespread access to lutetium PSMA in many countries outside the US. So that's already become almost a de facto standard of care without the evidence. So
then I have one question for you all. Yeah. My question is, then in the control arm of Vision, what percent of people got lutetium PSMA post-protocol?
Yeah, I don't know that. Probably not,
maybe
because
it would be very high then, arguably.
You would have to fly outside the USA because it was not available in the US. Yeah. And in our experience, most people living in the US think it's the best country in the world. So they don't leave. They don't seek therapy in India.
But if you ran that trial...
Don't worry. With COVID-19, we proved we're not.
But yeah, it's a good point, isn't it? But I think it probably is worth a correction on it because this is why we have widespread access to both PSMA PET-CT and lutetium PSMA in very many countries outside the US. And that's just the way it is. It's become a de facto standard of care. And it's, to me, Vinay, it's been interesting learning a bit about the radiopharmacy radiopharmaceutical industry and nuclear medicine over the years that, I mean, it's partly part of the allure that these clever radiochemists in Germany are mixing up these peptides and creating this stuff and put the formula on the internet, and then here in Melbourne we crank it up the next day and stick it into a patient with some sort of regulatory exemptions. But it's part of the beauty. That's how they innovate. But also, as Michael pointed out earlier, it's part of the issue as well, because then you get this compassionate access use and it becomes widely available and it's disruptive and so on. So we need the mixture of the innovation that the radiopharmaceutical industry and their radio chemists can do with some good trials still. Is that right?
Yeah, so we could have a whole session on nuclear medicine and the FDA, because what's happened in the US is it's very hard to use these compounds in humans before you have your phase 1, 2, 3 data, because you don't have those German Australian compassionate access laws. And that's good and bad. It forces you to do clinical trials, which we all support. But if you're going to do them badly, then big deal. But it does freeze that early development, because you need a few $1 million to do those first trials, whereas in Germany and Australia, you can play with that quickly and develop something novel that's good for patients. And I think we need a bit of a mixture. So the FDA has sort of both sides. I think they've frozen the early development of some of these compounds, because you've got to be an industry, to get a compound off the ground. And then when it comes to the phase 3, they're, not having the optimal trial design. So it's a double whammy.
I guess the only thing I'll add to that is that if you're able to do it at such a low price, I commend you for it. That's, I think, a really remarkable thing. And I guess one of the things that it makes me think about is chimeric antigen receptor T cells. I think it is now a commercial drug product at $400,000 a bag, but there was an opportunity to actually turn it into a cellular product like we do bone marrow transplants. And in our labs at any university, we can, increase the certain CD4 ratio to CD8 or CD34 and to treat it like that. And that would have been way cheaper had we done it that way. So I think, yeah, it's a very interesting point. Yeah. I don't think I knew all that about radionucleotides.
There you go. Have we taken enough time? What else?
We could talk forever about
I know
this.
we could, but some of us have
one
jobs
thing
to do.
I wanted to quickly bring up, Michael, is that, especially after ASCO, one thing the Vision trial has prompted is, there's now a call just to treat all comers with this, and instead of doing PSMA PET screening.
Yeah, we should touch on this,
and that's
it's an
something I know
important
you
topic.
feel very strongly about.
So when you see that response rate in the therapy trial, 66% compared to 37% with cabazitaxel, big difference between the two arms. We selected patients very carefully with PSMA and FDG PET. So only patients with high PSMA expression at the sites of metastatic disease were included. So we excluded 30% of patients in the therapy trial. Now, we think that's good. We're excluding patients that aren't likely to benefit. It's like selecting ER-positive breast cancer for estrogen receptor-directed therapy, entirely appropriate. And the Vision trial did the same. They did a PSMA PET, and 12% were excluded. They did not do the FDG PET, and they had a little bit lower threshold for the PSMA PET, reasonable for the type of trial they were doing. And in the commentary post-ASCO, it was like, well, let's not select patients at all. Let's just give lutetium PSMA to everyone, and then your response rate's just going to drop even lower, and you might not, we don't know the results of that. So it'd be interesting to see what the FDA comes up with here as well. Are they going to mandate a PSMA PET to select patients with high PSMA expression, or are they going to say, oh, look, almost 90% was suitable in the Vision, you don't need any selection, it's a good treatment for everyone?
I suppose there's an expedited review going on, and they tend to keep the label pretty close to.
Point. Yeah. I guess the only thing I would add to Michael's point is I think the way Michael did it is better, but the way Vision does it is bigger market share.
Correct.
I think we'll all agree there, right?
Yeah. And clearly, and even in the discussions that the authors have done since, you can clearly say we were commercial this and want to be pragmatic and have a big pool, et cetera, et cetera. I get it. And, there's some value in that. Good. I think we should
I feel like we've held Vinay back a bit. Vinay, maybe we should give you 30 seconds to do a rant.
I guess here's, all I'll say in closing thoughts on this topic is, you didn't ask me about my clinical practice, but my clinical practice has always been broad. I'm in my 6th or 7th year on faculty. And over the years, I've worked at places where, every time some faculty member left for whatever reason, I inherited a portion of their patients. So I've taken care of solid tumors and heme malignancies, and my practice has always been broad. But the core of what I do, and actually currently I'm in the Department of Epidemiology as my primary appointment. I still, of course, attend for 3 months a year, and I run clinic, but I'm an epidemiologist. And I guess what I would say is that, it's wonderful to talk about this trial, and I think it raises so many of these issues, but these issues, I think, really do span all of the trials. And every time you read a randomized trial in our field, there's just a few questions I ask. One, is the endpoint that really matters to people, living longer, living better? 2, is the control arm what you would have done if you really had that person in your clinic? 3, after they progressed off treatment, did they do what you would have done had you seen that person in your clinic? 4, always look at dose reductions. Sometimes these novel drugs, they push the dose and they don't do it in the control arm. So there's a difference in dose reduction. And I think that those will cover a lot. It will really kind of show that many of these trials don't really answer the question that we face as doctors.
Fantastic.
Vinay.
Keep asking the hard questions, Vinay, and thanks so much again for joining us. Well, that's all we've got time for today on GUcast. Thanks very much, Michael, for joining us. Great to see you again. And Vinay, thank you for joining us. We'd love to have you back on the program again. Keep up the good work. We love listening to Plenary, and congratulations.
We can go to the pub.
I know, we can go to the pub today. Hooray, it's Freedom Day in Melbourne, and that's exactly what we're going to do.
I'm going to the hairdresser.
Are you?
Yeah.
No smart comments.
I need.
Thanks so much.